Okay, well, welcome everybody. Today is Nobel Prize Day in Stockholm, and we've planned a very special event to highlight it. Nobel Prize ceremonies are always held on December 10, the date of Alfred Nobel's death. The award ceremony started in the late afternoon in Stockholm, which is mid-morning in Nashville. Today is the 35th anniversary of Stanley Cohen's 1986 award. So it's a great treat to have Jan Jordan with us today to share her remembrances of the award and their trip to Stockholm. Stanley's body of work and his prize is his legacy to us. We feel a special bond as members of his department. And in fact, there are a few of us who were here as his colleagues while he was still working. Stanley is not only someone to be proud of, but someone to emulate. He exemplified the scientist who could identify an important problem and explore it with a singular dedication. He also did many of the experiments himself. He was also someone who collaborated with colleagues to extend the impact of his research, but remained the driver of the program to maintain focus. I remember at a biochemistry retreat, a student asked Stanley if he ever felt he had gone down a blind alley in his research. Stanley replied that all he cared about was how EGF worked. So as long as he was pursuing that goal, he didn't feel he could go down a blind alley. Many of us were blessed to have Stanley as a colleague and were sad when he retired. His words to Mike Waterman to explain it were, the literature has gotten so big, I can't keep up. And if I can't do that, I can't do good experiments. Stanley lived a rich life with his wife, Jan, both before and after he retired, and enjoyed living in Tucson and then back in Nashville. Although he developed Alzheimer's disease later in life, he lived to the ripe old age of 97 before passing away last February. Stanley had a massive impact, and we were delighted to honor him by creating the Stanley Cohen Innovation Fund for high-risk, high-reward research. I am so grateful to Jan for sharing the excitement of the Nobel Prize and their trip to Stockholm. Jan grew up on a small farm south of Franklin. She attended Franklin High School and was a three-time all-state basketball player. She attended Vanderbilt University and received a degree in molecular biology. While here, she played on the first Vanderbilt women's intercollegiate basketball team as a forward. After college, she took a job working in a basic science laboratory and continued working in basic science labs for 25 years. She met Stanley in 1979, and they married in 1981. We are truly blessed to have her with us today. The title of her talk is Retrospective on Stanley Cohen and Presentation in Sweden of the 1986 Nobel Prize in Physiology or Medicine. So
get me.
Yes, I think this is where you want to be. Yeah. All right. Of course, now we have a problem. Let's see. Let's try this again.
Okay. Thank you, Larry. It's a pleasure to be here today. I was just telling Larry that I haven't been on campus other than for doctor's appointments for literally years. So it's nice to be back. Just some history on this presentation. Back in 2013, Stanley and I lived out at the Heritage at Brentwood, and some of the residents came up to me and they said, Jen, can you do some kind of presentation on the Nobel Prizes? So I sat down and I thought about it, and then I said, well, this is going to have to be a slide presentation. So I had to learn Keynote, Apple's answer to PowerPoint, get all the information together, photos. So it took quite some time to put it together. So I gave it there. It was a big success. I gave it at the Franklin and Brentwood Chamber of Commerce. I gave it to in the community where I now live, Brookdale Belmead, and then I went to Brookdale Corporate and gave it there, and that was a big success. And so now I'm giving it here, and I appreciate you all being here. the talk, the first part is about the prizes, the origin, how they're selected, some interesting facts. Then I'm going into some science, which I know is going to be like preaching to the choir, but I just felt like I had to go over a little bit of that. I was reading the literature while I was preparing this, and the amount of literature in this area is absolutely overwhelming. Then the last part of the talk, which I think most people really like, is about our trip to Stockholm. I have a nice video in there that I edited. So anyway, to get started, here's Stanley. He's the reason that we're here today. He was a smart guy, but most importantly, he was a kind person and a very humble person. And so there he is. Okay, Alfred Nobel. Nobel was the Swedish chemist who invented nitroglycerin and dynamite. He was educated in Europe and the US. He was an interesting fellow. He never married. He had 3 significant relationships with women, 2 that he proposed to, but they didn't accept his proposal. And then a 3rd one who he corresponded with until his death. He had some 2 or 3 residences in Europe, and he would kind of go back and forth between those residences. He had armament plants all in Europe and the US. And his brother, Emil, died in an experiment with nitroglycerin, and a paper in Stockholm ran an obituary, but they ran an obituary on Alfred Nobel, not his brother. And one of the sentences was, "The merchant of death is dead." So obviously the paper didn't like Nobel making all of his money off of armament plants. And so Nobel read this, and he said, it troubled him. And so he sat down and he rewrote his will, and he put his 675 million, that's present day dollars, into a fund. And he wanted the interest of which will be used to endow prizes to those who, during the preceding year, shall have conferred the greatest benefit to mankind. So the areas that he wanted the prizes given in was physics, chemistry, physiology or medicine, literature, and peace. Now, one of the women that he was involved with, Bertha Kinsey, she was a big peace activist. And so folks think that that was one reason that he established the Peace Prize, was because of her. The Economics Prize, a bank in Stockholm gave a large sum of money to the Nobel Foundation so there could be a prize in economics. It's the prize of economic sciences in memory of Alfred Nobel. So that's given at the same time as all of these other prizes are given in Stockholm. Okay, so the Nobel Foundation was established. That looks after everything. Most importantly, it looks after the investments, but looks after the ceremonies, getting the laureates to Stockholm, just everything the Nobel Foundation looks after. These other groups here, they choose the winners. And the Nobel Assembly of the Karolinska Institute, it chooses physiology or medicine, and that's just the medical school there at the Karolinska. Now the Norwegian prize, the Peace Prize is chosen by a group in Norway. There are 5 members of that group, and they're supposedly very independent and don't pay attention to anything, any outside influences or whatever. Of course, it's usually can be somewhat controversial. Okay, the nomination process for physiology or medicine, this is probably the same for chemistry and physics and probably economics, because these come predominantly from universities. So nomination forms are sent out to the Nobel Assembly of the Karolinska Institute, to past Nobel laureates, and to faculty of outstanding medical schools throughout the world. And Vanderbilt's faculty, they received these nomination forms. And in fact, one of the labs I worked in, David Garber's, he was a scientist here. He was a great guy. He was a really smart guy, but he told me that he nominated Stanley for the Nobel Prize. This was many years after Stanley had won. So did Stanley know he had been nominated? Well, no, because the process is secret. So he didn't know. But there's an award given each year, the Lasker Award, and 75% of the winners of the Lasker Award go on to win the Nobel Prize. And Stanley won the award that year in 1986. Just a couple other comments. You can't just write a name in on the nomination form and send it in. You have to be able to tell and describe why the work is important, why it's groundbreaking. And so it takes quite a bit of effort to do that. And Stanley, I mean, his work, scientists knew that his work was important. There were comments like it's prize-worthy, and so anyway. Okay, 4 people have won more than one Nobel Prize. So one's not enough. They had to win more than one. So Marie Curie, she won in 1903 with her husband, Pierre, for the discovery of the elements polonium and radium. And Pierre was killed in a freak street accident. And she went on to win the Nobel Prize in 1911 for the final purification of the element radium, and then for some work on the phenomenon of radioactivity. And she actually helped set up mobile X-ray units that were used in World War I. She was born in Warsaw, and back then, of course, women couldn't go to universities, but there was something called Flying University, which was basically an underground university for women, and she went there, and then she followed his sister on into Paris, and her career got started. Frederick Sanger, he won the prize in chemistry for elucidating the protein structure of insulin, so that was pretty important. And then he developed a method for sequencing DNA, and he won the chemistry prize again for that, and that was kind of a linchpin in the molecular biology revolution. And in fact, I actually used that method probably hundreds of times to sequence DNA. Linus Pauling, he wrote a paper, The Nature of the Chemical Bond, and later wrote a book. So that was very important in that area, and he won the chemistry prize. And then later on, he was a big anti-nuke person. So for his work in that area, he won the Peace Prize. And he was also the guy who said that vitamin C is good for colds. John Bardeen, he won in physics for developing the transistor. So that was important because then you could miniaturize electronics. And then later on, he won a second physics prize for the discovery of superconductors. Okay, so these are just some interesting facts about the prizes. The first Nobel Awards were given in 1901. So that was, he died, Nobel died in 1986. So they were able to get this all together in 5 years. There have been 934 laureates, only 57 women. So this is kind of a measly number of women. of course, way back, women were discriminated against and certainly not encouraged to go into the sciences. NPR did a program about this and said that the Nobel committees were old boys clubs. So I don't know if that's true or not, but they have decided, the committees actually publicly came out and said that when they send out the nomination forms, that they are asking the people to consider diversity in their nominations. But that's still a really, really tiny amount of women that have won Nobel Prizes. The award money today is $1 million. Believe me, in 1986, it wasn't anywhere near there. And in fact, we probably spent all the award money bringing our families over to Stockholm. The Nobel laureate's average age is 59. Stanley was 58, so he was right on the money. The money that's in investment is exempt from taxes in Sweden and also exempt from investment taxes in the US. The youngest winner was 17, Malala Yousafzai. She was a young Pakistani woman who was encouraging young Pakistani women to get education, and the Taliban shot her in the head and she survived. So she was the youngest winner at 17. And the oldest was this guy, Arthur Ashkin, who won for the development of lithium batteries, and he was 97. Okay, so Stanley's academic background: he was born in Brooklyn. He attended Brooklyn College, where he was able to go for free, because back then, if you had a certain GPA in high school, then you could go free to Brooklyn College. And if it hadn't been for that, then he never would have been able to afford to go to college, and we never would have had Stanley winning the Nobel Prize. He received his master's in biology and chemistry from Oberlin College, and then he completed his PhD in biochemistry at the University of Michigan. And his dissertation was nitrogen metabolism in earthworms. So he joked that he probably collected 1,000 earthworms. He'd go out at night and collect earthworms and for his dissertation work. He did 6 years of postdoctoral work at WashU in St. Louis, and then he came to Vanderbilt in 1959 as an assistant professor and progressed on up through the associate to full professor. Okay, why did Stanley win the Nobel Prize? So I know I'm preaching to the choir here as far as science goes, but I just felt like that I needed to address it some. And it also gave me an opportunity to read the literature and figure all this out. So Stanley was working at WashU in St. Louis with a woman, Rita Levi-Montalcini, with whom he shared the Nobel Prize. And he observed that when a crude extract from mouse salivary glands were injected into newborn mice, he saw that there was early eye opening and early tooth eruption. And I remember somebody, it was in some lecture, but somebody asked Stanley, how did you know this was important? Well, this was where Stanley he was a smart guy, and he knew a lot of different areas: biochemistry, chemistry, cell biology, embryology, whatever. But he answered, he said, I thought that anything that speeded up a natural process must be important. So when he came to Vanderbilt in 1959, he studied this. And this led to the discovery of a protein called EGF, epidermal growth factor. And then he also isolated the epidermal growth factor receptor. So what he observed was that when EGF bound to the EGF receptor, and this is the receptor here, just a schematic, that in the intracellular region, the receptor was phosphorylated. So Tony Hunter had just found out that Rous sarcoma virus has tyrosine kinase activity. So Stanley looked at that, and sure enough, there was tyrosine kinase activity with the EGF receptor. And another thing that he discovered too is that EGF, the ligand, bound to the EGF receptor and was internalized into the cell. So previously, peptide hormones, they were thought to bind to the receptor, do their thing, and then be released back into the extracellular portion of the cell. So Stanley, he discovered two things: a new method of hormone action with the tyrosine kinase phosphorylation, and then also the binding of the ligand in the internalization of the receptor ligand into the cell. So this was so important biologically. I mean, other receptors, they were found to work the same way, that he was awarded the Nobel Prize in Physiology or Medicine in 1986. So now the EGF receptor pathway is one of the most important in mammalian cells and is also one of the most significant cancer pathways in cells. And this is just another schematic. The receptors, they form a dimer, they dimerize, and these just show the phosphorylation sites on the receptors, and adapter molecules and proteins bind to that or take the phosphate off, and this begins the signal transduction pathways in the cell. And this just shows the pathways that occur when EGF binds to the EGF receptor. You have normal cell proliferation, growth, differentiation, migration, and regulation of apoptosis. But then when things go wrong, you get invasion, angiogenesis, metastasis, evasion of apoptosis, and then bad things happen with the cells. So there are now clinical applications of Stanley's work. Some of the following cancers have mutations in either the extracellular portion of the EGF receptor or the tyrosine kinase. And there are currently 87 drugs on the market for the treatment of cancer based on Stanley's discoveries. Okay, so the regulation of EGF receptor signaling, there's several ways that the signaling can be abrogated. It can be messed up. And this is a few of them here. This is simplistic, I know, because there's so much information. But you can have enhanced production of the ligand. So there's several ligands that bind to the EGF receptor, but primarily it's EGF and the transforming growth factor alpha. You can have overproduction of the EGF receptor protein. You can have mutations leading to constitutive activation of the EGF receptor. So there's a mutation in the extracellular portion of the EGF receptor where that part is truncated and there's no binding of the ligand, but the dimer still remains active. There's also deficiency of the EGF downregulation where it's supposed to be, goes through the disposal process into the lysozymes and it's degraded. And then there's mutations leading to cell cycle progression through transcription factors in the cell nucleus. So EGF was found to be present in the nucleus. It's translocated into the nucleus. It acts as a transcription factor or a co-transcription factor. So here it's a transcription factor for cyclin D. is a co-transcription factor in these three instances here. And all of these lead to, or can lead to, tumorigenesis, proliferation, metastasis, chemoresistance, and radioresistance. Okay, so the mutations in the EGF receptor extracellular and tyrosine kinase domains. The mutations in the extracellular domain are usually overexpression of the EGF receptor protein and then a constitutively active dimer with no ligand binding. The tyrosine kinase domain mutations, again, the constitutively active dimer, they drive the cell through cell cycle progression and produce an anti-apoptotic pathway. Okay, so some drugs that have been developed, monoclonal antibodies were the first ones developed. These inhibit cell growth, induce apoptosis, and decrease production of other cellular factors in the signal transduction pathways in the cell associated with tumor progression. Tyrosine kinase inhibitors block the phosphorylation of the kinase domain and have the same effects as above. Okay, so monoclonal antibody drugs, here we go here. The first one that was developed, Axel Ulrich out at Genentech, noted that there was an overexpression of the EGF receptor in certain breast cancer cells. And so Genentech developed the first monoclonal antibody drug for the treatment of mutations in the EGF receptor. These tyrosine kinase inhibitors, there's first, second, and third generation. I think there may even be fourth generation now. One reason that they have to keep making these tyrosine kinase inhibitors is because after a while of treatment, the cells develop a resistance to these tyrosine kinase inhibitors. And some of these are caused, like one is a secondary mutation that develops with the treatment. Then there are other mutations that occur in the EGF receptor, which result in this resistance to the tyrosine kinase inhibitor drugs. They're especially known in the treatment of non-small cell lung cancer cases, and that's why they were first developed. They're now used, I believe, some in glioblastomas, just like monoclonal antibodies are also used in some glioblastomas, some colon, some head and neck, just several different cancers. Okay, so this is, I guess, kind of the fun part. This is the announcement and our trip. So you probably heard that the winners get like a 3 or 3:30 in the morning call from Stockholm to tell them that they won the Nobel Prize. So the phone rang one morning about 3 or 3:30, and Stanley and I looked at each other and we going, oh no. And so he went to answer the phone. He came back and he said, well, I just won the Nobel Prize. So I was like, oh, okay. And this is no joke or no lie or whatever. He went back to bed and slept for like another hour and a half. And then until the media started calling, and he just had to get up. So this is a picture down at Vanderbilt at the press conference. I like it because it's a good picture of Stanley and me, and with me in the background. And Dean Chapman, the dean of the medical school, after it was over, he came up to me and he said, you know, Jan, he said, we're happy that Stanley won the Nobel Prize for Vanderbilt, but we're also happy that he won personally because he's such a nice guy. And I really appreciated him coming up and telling me that. And I think that's generally how people felt about Stanley. So this is the press conference in Stockholm. There's Rita Levi-Montalcini. She and Stanley, they were very close. They worked together. I remember Stanley said once, Rita knew nothing about biochemistry and I knew nothing about embryology, but separate we were good, but together we were marvelous. So they had a really good relationship. She was I just have to say, she was quite a woman. She decided early on that she was not going to get married because she wanted to dedicate herself to science. And, you know, as a woman, you sort of have to do that, but not necessarily, you know, as a man. And she was a very interesting person, and she was also a kind person. Okay, so the main events during Nobel Week, the 4 main events are the Nobel concert, the Nobel lecture, the prize ceremony and banquet. That's the big one. And then the banquet at the Royal Palace. This was Christoph, our attaché. He made sure we got where we were supposed to be, when we were supposed to be there. And we really needed him because we were gone from morning to night. And this was Guy. He was our attaché, our chauffeur. He was also our security guard because he carried a gun. It happened that 6 months earlier, their prime minister had been assassinated, and violence was kind of unheard of in Sweden. So they were very, very cautious about their laureates that year. This is the hotel we stayed, the Grand Hotel, the lobby. Lori and I, Stanley's granddaughter, we used to come down and play cards with Guy, the chauffeur, until everybody was ready to go to the next event, and that was a lot of fun. This is old city Stockholm. We spent a whole day there wandering around, and it was nice. I really liked old Stockholm. This is just another picture there with the boats and the narrow streets. This is a group picture. It's kind of pixelated, but that's my mother and Bert, Ethel, Stanley's sister, me, and then Lori, Stanley's granddaughter. So that was our whole group there that went to Stockholm. This was just a beautiful synagogue that Stanley and Rita went to. Rita was Jewish, and of course Stanley, with the name Cohen, that's about as Jewish as you can get. He wasn't a religious Jew, but they visited one night there. This is Stanley giving his Nobel lecture. You have to give a lecture before you can get the prize. But it was sort of funny because there was 2 hours allotted for Stanley and Rita's lectures, and Rita just kept going and going and going to an hour and 20 minutes. And then finally Stanley could get up there, but he really had to rush through it and eliminate certain slides, but he wasn't concerned. So you have to practice the ceremony before the ceremony. So there's Rita and Stanley there on stage. That's Wally Soyinka. He was the literature winner. He was from Nigeria, and he had to get special permission to wear his native costume to the ceremony. So here we are, the night of the ceremony and the banquet, and I was much younger then. In fact, I was only like 31 at that picture, and that dress, that set us back about $600, but you know, you have to do it. This is just a photo looking down on the stage where the ceremony takes place. this wasn't the night of Stanley's ceremony. It's just one that I found. Okay, now this is about a 5.5-minute video. I actually downloaded a video from the Nobel Prize website and edited it. Unfortunately, in 1986, you didn't have the videos all over the internet like you do now. But I chose the year 2000 because I knew one of the winners, Eric Kandel, who won for his work on memory. And the reason I wanted to show you this is because this is a beautiful ceremony. Now I know that, I'm more emotionally involved with it, but I think you can see what a beautiful ceremony it is. The king, the royal family, they come in. Well, there's the announcer guy talks, and then the royal family comes in, and then the Nobel laureates come from the back of the stage, and they come in. And then I use a couple examples of the winners receiving the prize from the king, just to give you an idea of what it's like to receive the prize from the king. And then at the end, they play the Swedish national anthem at the end. And I think that it's beautiful. I hope you'll enjoy it. You have to keep in mind that, of course, I was very, sort of emotional, but I hope that you enjoy it.
Good afternoon and welcome from Stockholm. In a few minutes, we shall be joining those who are inside the Stockholm Concert Hall as we witness this year's Nobel laureates receive their awards from the hands of His Majesty Carl Gustaf XVI of Sweden. Queen Sylvia and other members of the royal family of Sweden will be on stage together with this year's laureates. Also on stage are members of the prize awarding institutions. Among the audience are family members of the laureates, representatives of the Swedish government and parliament, and other guests, including some lucky students. And now the royal family are taking their seats in the hall. As you can see, the audience rises for the arrival of this year's Nobel Prize winners. And finally, of the three, Professor Eric Kandel.
现在，我请你接受瑞典国王陛下亲手要颁给你的
And you may not realize, but in fact, the professor is addressing this year's winner in his own language, in Mandarin Chinese. At this point. So Gao Xinjiang steps forward to receive his award, this year's Nobel Prize for Literature, from King Carl Gustaf XVI of Sweden. The national anthem of Sweden, Du gamla du fria, to traditional music and a text by R. Dybeck. Here you can see the assembled members of the Swedish royal family, many of them singing the national anthem. Here's Princess Victoria.
So I hope you enjoyed that video. I like to watch that. This is all the winners for Stanley in 1986, the year he won. These are chemistry guys, physics, chemistry. Stanley and Rita, Wally Shoyinka, James Buchanan. He's interesting. He went to MTSU, and that was college for teachers. And then he went on up to the University of Chicago and started his career in economics. This young guy, physics guy, I'm sorry, I don't remember their names, but he was only 35. And I remember Stanley saying, oh, he's so young and he's going to have to live up to this for the rest of his life. So I thought that was kind of whatever, interesting. Here's Stanley and Rita standing, getting ready to receive the award. And then that's Stanley receiving the prize from the king. This is City Hall, where we went to the banquet afterwards at night and during the day. Stanley and I were somewhere up here. We were proceeding down into the banquet hall. There are over 1,000 people at that banquet. This just shows your view down there. This middle table is where the king and queen sit, and Rita got to sit next to the king, and Stanley got to sit next to the king, to the queen. So that was sort of neat. Although I would have rather been sitting with Stanley, but anyway. This is just another view down the table. Here's the dessert procession. They made a big deal of the desserts. This is just the place settings there at the middle table. This is me and my mother getting ready to go to the Royal Palace for the banquet there. And there's Stanley and me. We look pretty happy. This is the Royal Palace at night and during the day. And this is the banquet, just this one long table with folks. Not too many people other than the laureates and their spouses. And then this is Stanley and me. We're at the last event of the 10-day period we were there, and we were happy, but we were also tired, and we wanted to, we were ready to go home. So here we are flying over the fjords of Norway, and then over the clouds. Just one thing about Stanley, I think if he were alive today, I mean, for one thing, Stanley's research just shows that basic science research is important, and that high risk basic science research should be done. But I think if Stanley were alive today and he could look at all of the literature, and he would keep up with things. And I think sometimes occasionally he would sit down and just smile. Okay, just on another personal note, Stanley and I got really involved in jeeping. We lived out in Tucson for 10 years, and it's kind of weird. You wouldn't think we would, but we were really into jeeping. We went on all these off-road trails. We had this Jeep modified. It could go anywhere. And I did the driving, and Stanley did the spotting. We went to these Jeep jamborees, and we had a great time. Marlene Jane, who was administrator in biochemistry for, I don't know, 30 to 35 years, she would come out and visit us in Tucson, and we go on all these Jeep jamborees, and she's such a dear friend. I was hoping she could make it down today, but she's here via Zoom. And this is just us up in Moab, Utah, going over a trail. And here we are. You can see we go over some pretty big rocks. And here's another obstacle. And then there's Stanley. So thank you very much for tuning in. I've enjoyed giving this presentation, and I thank Larry Marnett. And so thank you. Hope you enjoyed it.
Okay, well, thank you, Jan, for a really great presentation. This was really fun. I think we've got a few minutes for questions if people have them. Maybe just either speak up or raise your hand. It's a little hard to see everybody. Any questions at all? Connie Wagner, I see you on there. Hello.
Oh, Connie. Hey, Connie.
I think you're muted, Connie.
There you go, Dr. Wagner. Now, tell them hello.
Hey, Connie.
Hey, Jan, it's just a pleasure and to see you again. I haven't been able to get around very much, but you look great. I hope you're feeling great. And
Yeah.
Great. And so I remember when Stanley called me up, he says, I'm getting married today.
Yeah, I remember that too.
He wondered if Jane and I would be could be at your wedding, and Jane said, "Oh, that's great. Do you have a ring?" He said, "No."
Oh, that's true. It was a justice of the peace thing, Connie.
Yeah. Well, it's been wonderful seeing you again, and I can't travel very much now, but I appreciate what you've done to tell people about Stanley. He was a good friend. We played tennis twice a week. And you remember you played with us too.
Yeah, I did too.
Okay, so
Okay.
thank you again. It was very enjoyable and very educational.
Yeah, thank you, Connie.
You're welcome.
Good to see you.
It is.
That was awesome, wasn't it?
Well, there's Marlene.
Yeah, you wanna say something to Marlene? So we see Marlene Jane in there as well. Marlene, you want to say anything or?
You have to unmute, Marlene.
There you go.
Okay. It was a wonderful presentation, Jan.
Thank you,
I
Marlene.
enjoyed it so much. We had a ball. And
Yeah,
it
we
was-
did.
I mean, we went all over the country, from
We sure did.
California to the Outer Banks.
Yeah.
I didn't know much about science, but I did know the administrative part. And I
Yeah, I know you did.
trouble. And I was at Vanderbilt for 43 years, dear.
Okay, I was a little off then. But not all of that 43 was in biochemistry.
Yes, it was.
Oh,
I
I
had-
thought
I
it was someplace before.
I had 2 years in economics,
Oh, okay.
and I had 43 years in biochemistry in the same department.
That's unbelievable.
6 chairmen and Stanley.
Yeah.
And all of my professors, I feel like they were all mine. I still feel the same. And I'm looking forward for you to come out and visit me out here.
Yeah,
And we have lots to talk about.
yeah, we do. Lots of good memories.
Oh, yes. Oh, yes.
So there's
Thank
something.
you for doing this.
Let's see if it's in the chat. Okay, just some thank yous in the chat room. If there aren't any other questions or comments, let's just thank Jan for a very special presentation on an important day for the department. Nobel Prize Day always means something at Vanderbilt this time of the year in biochemistry. So thank you very much.